So welcome everybody to our fall 2022 water monitor meeting. And I want to say thank you everybody for showing up. I'm going to hopefully be hearing from a lot of other people besides me, which is always much nicer. I guess before we get started, I just wanted to say if anybody wants to say hello, introduce themselves, or anything like that, feel free. We'll also have a Q&A where people can bring up questions, issues, anything like that at the end as well. So if nobody especially has any announcements or anything they want to make at the beginning, then I'll just go ahead and start. I've got a screen I'm going to share here. And that's not the beginning of it. That's the beginning of it. So again, welcome to our water monitor meeting. What we've got on tap for this evening is I want to talk a little bit about new technology for bacteria that we're starting to try out, and it looks like it's going to be the way to go. I want to hear from our different programs. So I've got different people have said that they would give a little update about what's going on in their program, and I always think that's super interesting to hear. Gary Olson is going to talk a little bit about the DNA project that we've been working on. And anybody who has stories, issues, questions, anything to share, I'm going to highlight a brief one of a handful of new videos that were produced for us by our partner Cascade Water Alliance. And I think that's pretty much it. So just to jump in and start, the new bacteria monitoring technology that I'm talking about is something called R cards. And this is a little bit different than what we've been doing in the past, where previously we were using a technique called Colascan Easy Gel. And so it involved using, besides pipettes, you used little bottles of media and used Petri dishes. And there was a fair amount of waste, not to mention it took up a lot of space in your freezer. And so this new technique is a little bit easier to use, and it's also smaller, at least space-wise. So as I mentioned, it's easier to store. So you get a pack of these tests that's about the size of a deck of cards. Less waste. We're not dealing with Petri dishes, and conceivably we really don't even have to use sample little bottles anymore. We have been having some issues, some kind of quality control issues with the Easy Gel, so we get to move away from that technique. And now on the Global Water Watch database, they have a data entry option for the R card. So that is that's been looks like the direction we're going to go in. So in terms of different types of cards that are out there, if you look on it's a Roth Bioscience website, they have all different types of tests that you can do. You can do E. coli, coliform, you can do listeria, you can do all kinds of stuff. The primary ones that we're interested in, there's a E. coli, and you can get an E. coli only card, or you can get one that does E. coli and other coliform called ECC. In terms of sample collection, I ordered a packet of their complete sample collection, which comes with the cards and the pipettes and sample bottles if you want to use them. And you have different options on how you use it. So you can either pipette it directly at the creek and place a sample on the card there, or you can collect it in a container and place it on a card later, like say you get back to where you have a flat surface to put the card on. So you've got some different options of how you collect it. In terms of results, it's pretty straightforward. In fact, it's a lot easier because you count the blue dots and pink if you're using other coliform, and they give you this grid that just makes it really easy to see, and it's easier to see on this background than it is on counting from a plate. We're ordering some for monitors to use, and we're going to basically try them out and decide if it's going to be a permanent change, and then which cards we're going to do. I've talked with Serge at Global Water Watch, and they're using the ECC or ECCA, which has like a way to prevent getting a false positive color of an E. coli. But I think Gary Olson with Thornton Creek Alliance is just using the E. coli cards. So I'm gonna, that's all I have to say about those right now. I'm gonna stop sharing right now and maybe just kind of go. I didn't have a particular order to go in from partner updates, so I'll just say, if you are one of our monitoring partners, or I'll start picking on people, but maybe I'll just start picking on people and ask people to share. So maybe, how if I start with you, Anna?
Yeah, we just started using our cards and I love them. We have been actually reusing the sample bottles because we usually are sampling about 6 to 8 sites. So we do have to use the bottles to do that because we want to plate all at once. And I am trying to sterilize and reuse those bottles. I found a protocol for sterilization of the bottles online, but I'm actually trying to seek another one, a better one, if anybody has any advice. The only other update I would give, well, there's 2 other updates I would give. One is that we just put out our second water quality report that covers a 2-year monthly monitoring at Springbrook Creek Watershed in Kent and Renton. And that is going to be presented to the city councils in Kent and Renton this fall by the youth in our program with Unleash the Brilliance. So that's a big step for us to actually start presenting on our report. And lastly, I don't know if people saw this, but I didn't get this email directly. I got it after the fact, only like a week ago. But Ecology put out a call back in July asking for people to submit data for their next 303D list assessment. This is the list. This is the assessment of impaired streams. And I had never thought that Ecology was really accepting community science data, but it turns out that they do accept it. If there is a QAP, which this is why, Eric, I was asking about the status of the QAP for the Global Water Watch program. This is the Quality Assurance Project Plan that all research projects should have if they're doing water quality monitoring. And there is one for the Global Water Watch. I just don't know. I don't have it. So I'm trying to get it from Sergio. But I have learned how to upload data to the EIM, the Ecology's Environmental Information Management Database. And it's possible that if you have data that you think should be included in the assessment, I can put in the email address for the person at Ecology who is in charge of that database, and you could start a conversation with her and see if that's possible to still get that data in. The deadline was actually Friday, but it is possible to do. And I think that we're collecting good, valuable data that could be used in that assessment, and we should consider that. That's my update.
Just in case anybody doesn't know who you are, can you say who you are and what your project is that you're with?
Yes, I am Anna Bachman from Puget Soundkeeper. Sorry, it just says Puget Soundkeeper there. I'm with the Lost Urban Creeks Project.
Thank you. Well, that's exciting. That's a good opportunity with Ecology. Aaron or Eve, I don't know. Aaron, do you would just mention what's where you're from and what's going on with your group of monitors?
Yeah, hey everyone. My name is Aaron Hussman. I'm an education and outreach coordinator with the city of Kirkland. And then my colleague Eve Henrikson's on the call as well. If you want to introduce yourself to Eve. Maybe.
Yeah, hi. Sorry, I was changing my name to say Kirkland, so I couldn't unmute. Yeah, I'm an environmental analyst at the city of Kirkland and been working with our volunteers in the city. Yeah, we have had a couple of our volunteers monitoring at spots that there either was some recent restoration along the stream or there will be. So we're kind of hoping to track if there's any change in water temperature or any of the other parameters as that restoration grows up and shades the riparian area. And then we also just have some sites that are kind of just chosen to fill in data gaps places that we don't have time to monitor in our city program. But yeah, we have some great volunteers in Kirkland, so thank you.
Yeah, thanks, Eve. I wanted to just highlight Kirkland and Eve and Aaron, just because that's an example of a good partnership with a local government, with the local city, where originally they reached out to us and said, hey, we want to start some volunteer monitoring in our jurisdiction. And so we worked together to coordinate some training, and they rounded up some volunteers. We did some training. Eve helped us pick some sites, and now they've got some volunteers out monitoring in the community. So it's a good partnership.
Yeah. Yeah, and there was a couple things that if we have time later or whatever that I wanted to talk about, like some of the things that some of our monitors have noticed, such as sheens or suds, and like kind of how to tell the difference if they might be pollutants or not. So if we have time to maybe talk about that a little at some point.
Sure. Yep. Remind me, and at the end we'll kind of roll that into our kind of roundtable. All right. How about Kristen? What's going on with R2R?
Hi, everyone. I'm Kristen Dew. I'm an assistant teaching professor at UW's Human-Centered Design and Engineering program. I recognize a colleague here tonight that I've never actually met in person, I think, but I think I know your email, Sonia. So hi, nice to see you here. I've been coordinating the water quality monitoring on a project called Ridge to River, which is a variety of community groups and the Duwamish tribe, trying to understand the feasibility of connecting some somewhat disconnected trails in the green space around the Longhouse. And while that program or that project around developing trails is kind of going in a different direction now, and there are some changes in kind of leadership around that project, we continue to do water quality monitoring and try to figure out next steps for continuing that program in Puget Creek, whether with South Seattle College, kind of taking some of that on, or what that will look like, we're not sure. But we've been getting out about every 2 months for the last couple of years. Nothing super surprising or alarming came out of data from last year, other than that it was kind of surprising how high the dissolved oxygen was. I mean, these kinds of parameters that we expected to be kind of rough or terrible were not so as bad as we were expecting. So it's good to get another year of data going for that and share that out with these groups again. And hopefully they'll be continuing on next year as well.
Cool, thank you. And I remember just doing training with
monitors as well, so I will.
Just doing training with the Ridge to River group. We were doing some monitoring on Puget Creek in the Duwamish watershed, and we found kind of higher levels of alkalinity and hardness than what I typically find in other places, which potentially could be related to concrete kiln dust that was historically deposited throughout the area. So that was kind of an interesting finding.
Yeah, one that was expected, but yeah.
All right, thanks, Kristen. How about Brian? You want to talk a little bit about what's going on with the Stewardship Foundation?
Yeah, hello. I'm Brian Saunders, and I'm a biology environmental science instructor at North Seattle College and at Shoreline. And as Eric just mentioned, I'm also a board member of Lake Forest Park Stewardship Foundation. And yeah, we've got a good team now of 15, 16 people that are helping sample the Mackler and Lion Creek basins. In fact, this weekend we have our big day of doing our BB analysis, our yearly biological analysis. So that's always a fun time. And last year, just to kind of give you a quick update, we actually had a very significant find. We actually found a species called Synigmula, which is a flat-headed alderfly, and it's a very sensitive species. So we were really excited about that. So now we've kind of got this image of Sinigmula as our hope for the future for the creeks within our area. So we're trying to make a mascot out of it somehow, but it's taking some time for that. Yeah, so we're still doing our creek analysis, and we've just now switched to the R card. So we're very excited about that. We actually got it's very fortuitous in a way, because we had ordered the call scan and they were back ordered. They were going to take a long time. So we said, oh, can we cancel? Quickly. So that worked out. It was nice.
Thanks, Brian. Okay, I see David from the Karkik community. He's going to have to give me the correct acronym. CCAP, Karkik Community Action Plan.
Karkik Watershed Community Action Project.
There we go.
This is actually the Pipers Creek watershed. I don't know why they ever chose the name Karkik Watershed, but I guess because more people know where Karkik Park is. So yeah, I'm David Kuhn. I'm on the board for CCAP. I'm the director of salmon programs and the treasurer. I'm also on CPAC, which is the Karkik Park Advisory Council, which does trails work, forest restoration, and things like that in Karkik Park. And so we just now are starting to do or bring back the idea of monitoring water. We're mostly known, CCAP is mostly known for raising chum salmon in the spring and counting the chum salmon when they come back. Also working with SPU and parks on running the salmon stewards. So if you've ever been to Karkik Park, when the salmon are returning, there's people on the weekends talking to the public about it. We have people twice a week in counting the salmon and taking information on them. And see, what else do we do? We're part of the Salmon in Schools program, so we help support tanks in schools in Northwest Washington or Northwest Seattle. But anyhow, yeah, we've done water testing sporadically over our 33-year history, and used different methods, but usually it's been such a small group. And in the last couple of years, we've grown our volunteer base to the point where we have enough volunteers to actually do regular monthly testing at multiple sites. So right now we're targeting about 8 sites. And King County already tests in the lower reach of Pipers and on Pipers, with one at the lower reach of Venema. But they don't really test, they don't test at all any of the side channels or tributaries. So we've picked tributaries to test to see if any of the sub-basins of the Pipers Creek main basin are contributing or bringing in more pollutants than others. And this kind of came about, one, we've been wanting to do it as a community science program for some time, or at least I have for several years. But on top of that, we've had some test results from some of the sub-basins that have like alarming levels of copper, which I forgot I need to look into adding that to the GWW testing we'll be doing. But then you look at the King County data from down at the bottom of Pipers Creek right before it goes into Puget Sound, and they don't have significant levels. Well, that's because by then it's been so diluted with all the Pipers Creek and all the other tributaries that they're not catching what might be a higher source than what we should be expecting in the watershed from one particular site. So I look at and I also worry that there could be similar things, say with E. coli, that I think Eric the other day during the training mentioned a situation where TCA found a apartment complex that was leaking a lot of, basically pumping their sewage straight into the creek, and nobody really realized it until they were able to pinpoint it. So I'm hoping we'll be able to at least monitor for such things and for the impacts of some of the construction, as with much of Seattle, we're seeing a huge increase in construction within our basins. So hopefully by starting this program, getting some baseline data, we'll be able to monitor that. So anyhow, I'm excited that we connected with Eric and GWW. We've used other methodologies in the past with King County and/or the World Water Watch, which was not as rigorous, I don't think, or as frequent. So yeah, excited to be starting this new program.
Great, thanks, David. Yeah,
I
in
don't know if
fact,
there's any questions. Sorry.
yeah, and in fact, we just trained 14 new volunteers, I think 12 of whom are dedicated to Car Geeks program yesterday. So that was great. How about I've got Gary Olson with Thornton Creek Alliance. Gary, do you want to talk about what's going on with TCA?
Sure, yeah. So, oops, good timing on the phone call. Hopefully my wife will get it. So I'm Gary Olson. Yeah, I'm with Thornton Creek Alliance. I'm the lead for our community-based science program, and our program's been going since 2017. Thornton Creek watershed is the largest watershed in Seattle. Thornton Creek is basically two forks in the Northeast. It combined into one. And we've been testing about 37 sites over those 5 years, sometimes on a regular basis and sometimes just periodically, depending on what's happening in the watershed. In our case, we're primarily interested in E. coli. We share our data with Seattle Public Utilities, which puts our data into their database, which is one of the key drivers for our program, that we know that the information is getting used and getting observed by the folks that manage the creeks in Seattle. As somebody mentioned, there's been a lot of quality control problems with the EasyGel system, primarily with the Petri dishes most recently. And when we found out about the R cards, we said, hey, let's take a look at that. So the last month or so, we've been kind of doing some cross-checking between our cards and our traditional way of doing it. And our correlations have been reasonably good. There are some differences, but so far I'm pleased with it. And the one thing that Eric didn't mention about it is that there's a cost savings as well to using these cards. The other thing, which is kind of neat about it, is that they have a verification system. So you can order an additional small kit. And if you have a question about whether or not a colony is E. coli, you can kind of swab that colony after it's been incubated and then test it with a couple of other methods. And you'll get a positive test that confirms if it is E. coli. So that's kind of neat, because people that have used these systems before, there's often confusion about, is it blue, is it purple, is it teal, is it what, is it really E. coli? So this new method has that capability of doing that verification as well. So that's kind of neat. The only thing I'll mention, we have 3 schools involved in our program. And so actually they're just starting up with this school year. So we're going out and doing training of the students in those schools to start up their individual programs. And that's probably enough. I'll be talking about the DNA project a little bit later.
Awesome. Thanks, Gary. Is there anybody I missed, or anybody else who has some program or individual monitoring news that they wanted to share before we talk about the DNA project? All right. Well, Gary, why don't you go ahead and feel free to share slides or whatever you want to do.
Would you make me a co-host?
I made it so you can share.
Okay. All right. Now let me pull this up here. Let's say, there we go. Thank you. Okay, can everybody see that? Oops.
Yep.
All right, great. Let me know. All right, so the DNA project, we actually, Eric and I started talking about this project probably over a year and a half ago. So it's been a long period of time that we've been involved with really seeing whether this is something that we can get off the ground for community-based science programs. I spelled that alliance wrong, but that's too bad. Anyway, so DNA analysis. So there was a new technology developed within the last few years by the Lawrence Livermore Laboratories, where they developed what they call a phyllo chip, which allows them to analyze water-based microbes, bacteria, for example, and tie it back to the sources of those microbes. So that was kind of interesting. And then from their work, they established a company called Veriset. And the main researcher in this work, his name is Gary Anderson, set it up for basically commercial use to be used by various agencies that were interested in water contamination primarily. But when I talked to him originally, he was interested to see, if a community-based program would be interested in trying to use this to understand what was going on in their water systems. So the thing about this technology is it actually produces kind of a fingerprint from these chips to determine actually what the source of the E. coli are. So the way it's described is they can tell by these fingerprints whether the E. coli or the fecal waste that's coming in the stream comes from humans or from animals. And in many cases, specifically what animal it comes from, whether it comes from a bird or a dog or a cow or whatever. So it gives you kind of all the information. Anybody that's been involved with doing DNA analysis knows that traditionally you kind of have to run a sample specifically for what you're looking for. So if you're looking for human signals, then that's a test that you have to run, and it costs quite a bit of money to do that. So the idea of having all the information out of one test, which was probably, and I'll talk about the cost in a minute, considerably less than trying to get it for all of the potential things that could be in the water system. And that's what that last information is. So only one analysis needed to get complete information. So that's a definite cost advantage to this system. So when we look at it in terms of actual cost, so this isn't something that you would be doing on a weekly or monthly basis, but it's really after you determine that you have a particular problem or something that you want to delve into more deeply, then you would potentially take samples and submit them. So the way this procedure works, you actually filter through a very special filter, about 100 mils of sample. And those filters with the syringe, and that is roughly about $15 a test. These filters have to be kept at, once they've captured the sample, at a cold temperature of roughly 0 degrees Fahrenheit. And then when you ship them, you have to ship them in a thermal system, and it gets to be expensive. It costs about $300. So we generally build our samples up and send them as a group. And the DNA extractions, which they do for you, runs about $100 a sample. And then the actual DNA analysis is about $400 a sample. And they currently, as they're building their business up, kind of want to do it in groups of 16 samples. So if you send them a couple of samples, a couple of things can happen. One, they might charge you more if you're interested in getting your results back quickly, or they may hold them longer until they get other samples coming in to kind of get to that 16 minimum sample that's the most efficient for them in terms of running their DNA analysis. So just to give you an idea, obviously, Eric and I don't have the money on hand to do this. So we had to go out and look for that money. So we put a grant into the Waterworks grant program. And Rod Dembowski, our King County representative, who's really supported our programs over the years, thought that this was a valuable thing to do. And he eventually awarded us additional funds based on our current grants. And we went through the Sno-King Water Council grant as the main recipient of the funding, because Eric's funding for his program is significantly more than Thornton Creek Alliance's. But we also added some funding into Thornton Creek Alliance at about $5,000. And we're primarily covering the supply side of things and the shipping and those kinds of things. And Eric's primarily handling the big bucks, $400, $500 things. So anyway, so that's kind of where we are. And then also we do have some additional funds left over in our current grants to help us as well in the kind of program that we kind of laid out, which I'm not going to get into detail right now, but we have some things on that. So again, just looking at the purpose here of this particular grant is to determine the source of the high E. coli levels found in the creeks in our watershed. So that's the ultimate goal, because if we know what's causing it, we know where these locations are, but we don't know exactly what might be the cause. So by getting it more specific towards that, then hopefully we'll be able to reduce it or hopefully eliminate some of those sources. Determine if a community-based program can effectively use a new technology like this. Can we, as kind of non-professionals, if you will, and be able to do this work and be effective in it? And so that was another question that we kind of wanted to answer as we got into it. And then we started the program off thinking that we'd work with the King County Environmental Labs and do some crossover testing to see how their filtration and handling system compared with ours. And I'll talk a little bit about that in a few minutes. But also it allowed us to do a little bit of comparison of our E. coli results so that we could see how our Easy Gel systems were comparing with the county's method for doing E. coli, which is different for sure. And then finally, comparing filtration methods between King County Environmental Lab, which uses a nuclear pore filter, versus the Sterivex filtration system, which we're using for our DNA sample capturing process. So some of the requirements of doing this initial testing was kind of triplicate samples. So basically, for our community-based programs, our Eric's and Mark's, Eric uses a 1 mil sample, we use a 3 mil sample. So that was one set of material that we needed. The King County uses 100 mils plus, because they do kind of separate 100 mils and 10 mils and 1 mil kinds of sequencing for theirs. And then we needed 100 mils for our Sterivex filtering system. The samples had to go to the King County Environmental Lab within hours of sampling. So we had to coordinate that fairly effectively so that Eric got his samples, we got our samples, et cetera, and we got them over there, and then we did the filtration over there. Casey, the King County Environmental Lab had a homogenizing process, nothing technically fancy, but we had to follow the procedures that they had. And then filtering samples had to be stored at 0 degrees Fahrenheit or below. And finally, for those initial samples, we had King County Environmental Lab do the DNA extractions and do the shipping. So that was kind of the requirements that we initially had set up. And so these are the samples that we initially took for that comparative testing. And let's see, hopefully you guys can see the cursor is here. So basically we sampled in March, on March the 10th. These are all the locations. And I think we had, let's see, 7 or 8 locations that were in Thornton Creek. And Eric, I think you guys had like 3 or 4. Actually, I think we had 5 and you had 5. So that was the division for the samples that we took for the initial comparisons. And there's a little better chart that shows some of the values that we had that I'll show you next. Okay, so these are the sites actually from Thornton Creeks. And then they won't mean anything to you, but we give all of our sites site numbers. And these are the values that we got for our E. coli. So you can see that some of them were higher and some of them were low. So, and again, here's the samples that Eric had. And he only had 1 sample that was high. Most of the others were below 100. So we ended up choosing 5 of those. That's where those X's are. And those are the ones that we kind of split up in this triplicate package and had our initial sending off to Veriset after they went through the King County Environmental Lab process. And then down here, we didn't have 16, so we did some additional test sampling in the Thornton Creek Alliance area. And these are the numbers that we got for those samples. They were all fairly high. And so we sent those as Sterivex filter samples, and they did the DNA analysis on that. So the first thing I was going to show you is kind of the comparison of our E. coli analysis between the King County labs and Sno-King Watershed Councils and TCA Council results. So here's the results that we got. I just ordered them in terms of small to large. And this is the what King County Environmental Lab got on those. And that just plotted those out, but it has a correlation coefficient of about 0.8. So that's good. So that says that, between these techniques, we're getting reasonably good agreement. So it's always nice to have a nice verification of your method to a totally different method, but kind of giving the same information in terms of E. coli. So after quite a wait, we finally got a report back ahead of this meeting. So that was good. We haven't really had a total opportunity to go over everything on it, but I thought it would be good to share what we did get with you guys so you can see what kind of information that we've got. So basically, this is one part of the report, but basically the way that they're indicating. So here's the sample numbers here. And again, they're using kind of a different nomenclature that the King County Labs has, and then our site numbers. I'm not going to go into the detail of what these probes mean, but the phyllo chip has different kind of locations on it, and these probes kind of capture some of these fragments on them. So the numbers kind of relate to that. But what we're probably most interested in is what is the main source of those DNA fractions that are coming, that the phyllo chip is reading. And the way that they kind of look for significance, if the number, so down here, if you look at this, is what they call source tracker values. says if it's greater than 0.2, and they've kind of given it a red color, that indicates a strong source signal for what you're seeing. And then if it's between, if it's less than that, but between 0.5 and higher, then there's a good indication that it relates to it. And if it's lower than that, then it probably doesn't relate to that. So if you look at the samples that we had, for example, here's a sample number 2, and I won't go back to what it is, but just to give you an idea, there we've got a good, strong human signal. So that means human waste. And then there's this category they call ruminant. And ruminant is, they come back and say that could be an animal that pretty much chews its cud kind of thing. It's like a cow or a deer or something like that. They cannot distinguish the DNA signals from that between those types of animals. So just based on what they came back to say, they said, well, for us, based on those animals, you know, maybe that's a deer signal for you guys. I'm not so sure that that's the case, but that's kind of what they said. So there's some of the values that we've seen, pretty good human signal and this ruminant signal. And then out here, we've got birds and dogs and horses. We didn't, in the samples that we submitted, we really didn't see anything that was, you know, much over 0.1, which in their way of looking at it is not significant enough to count as being part of what that E. coli, you know, would be in the sample. So that's kind of the data. I'm going to quickly go through what our next steps are. So we want to try to understand what the data totally means. So we'll be working with Veriset on that. We've already had some conversations. Determine if any changes in procedures or handling are necessary so that we are sure what's happening and what's and what we're doing, and that we're doing everything appropriately. We now have some additional samples that we've taken through the summer. So we're going to be submitting those as well. And they're going to all pretty much high E. coli samples. Both Eric's got his in and we've got ours. And then we're going to look for other opportunities. If this is valid, you know, we definitely want to be able to share it. If we have funds, you know, to be able to do other folks, you know, key samples where you want to be interested in finding, you know, we'd be, you know, happy to try to make that happen for you if we have funds left over from what we're doing. So that is quick, but that's it for right now. And I'm going to stop sharing and take any questions anybody might have.
Thanks for sharing.
Oh, you're welcome.
Interesting information.
So Eve, I should tell you that one of the sites we did submit in the initial group was Firwood Creek. And
It looks like it got a deer hit.
yep, exactly. That's kind of what it looks like it got. But we can, I don't know what's been happening lately with the bacteria levels on Firwood Creek. We could certainly run another sample from that high.
Yeah, they've been through the roof lately if you want to run another one.
Yeah, so maybe I should go back and collect another sample from
Yeah.
Firwood.
One thing that I didn't mention to you is that in that classification of human samples, the interesting thing about this DNA analysis is that they can supposedly tell us the difference between whether the human came from a sewer system, meaning from like a house, whether it came from an encampment, or whether it came from
Septic.
Yeah, from a septic system, because they each have a different kind of fingerprint. So that's kind of fascinating and very kind of valuable. For this particular report, they didn't break that out. And so they're going to try to see if they can figure that out and maybe come back with that kind of information for us.
The other thing that we learned on our call today with Gary Anderson from Veriset was they don't have beavers in their library.
Yeah, right. They don't have beavers and they don't have crows. So anybody who finds some beaver poop or some crow poop, save it and we can send it to them and they can do their additions to their library.
So does anyone have any more questions for Gary about that project? Yeah, David chatted. I wonder if beaver are the unknowns, the high correlation unknowns. Yeah, interesting. Well, we'll have to collect some more
Yeah,
samples and maybe
that's-
add to their library.
Right.
Did you say how many do you have funding to continue sampling? I didn't hear what you said about
Yeah,
that.
so basically, so we have, roughly $30,000 plus some from previous grants. So basically, when you do, let's say you do a 16 run, so if they're doing the DNA analysis on it, you can say 500 times 16, that's about 9,000, roughly, yeah. It would be in that ballpark, $9,000 for a 16 run. So if we do we could probably do 3 of those kind of things. And what we want to do right now is do it through the seasons. So we'll have samples through the seasons. And then once we've kind of completed that, then we'll take a look at what we have enough funds to cover that. If we have extra funds after that, then we come back maybe to this community and or Seattle Watershed Alliance and say, hey, have you done E. coli and do you have a particular location or issue that you might want to add a sample to for a follow-up analysis?
I was also curious when you mentioned that there's a fingerprint for septic versus sewer versus homeless encampment. Aside from homeless encampment, I mean, do you have much septic in your study area, or isn't it all sewered?
In the Seattle area, it's primarily sewers, right? So my main interest would be encampments because we know we've gotten a lot of encampments in Seattle. So that would be my interest, but perhaps where Eric is, some of those areas may be more on septics. Or other people's areas may be more.
Kirkland has septics, some septics.
Yeah, right. So that would be
more likely to have that than encampment.
right. Yeah.
Okay, Eve, you said you had something you wanted to talk about briefly?
Yeah, and I mean, I guess it looks like not a ton of our volunteers are here. So maybe it'd be better to send it out in an email instead. I guess maybe most of our audience here kind of already knows the difference between suds that are natural versus pollutants. Is that something people are interested in talking about? I can just send out an email to our volunteers if that's.
I'd like to hear about it.
Okay. Yeah, so some of our volunteers, they're very observant and are making some good notes on their sheets, on their forms. And so sometimes people have noticed some suds in the water, and that's not always necessarily soaps or surfactants like pollutants in the water. Because sometimes, especially in the fall, decomposing leaves and stuff will kind of make a natural foam. And you can kind of tell the difference because the natural one is kind of more of a tan brown color rather than like a rainbowy bright bubble. It's like a more of a brownish color. So you can kind of keep an eye out for that. If it's natural, it won't have like a soapy smell either. Sometimes it's hard to smell it, but and then another thing that some of our volunteers have seen is a sheen on the water surface, and that can be gasoline or something, but it can also be a sheen caused by natural bacteria. And you can tell the difference if you kind of throw a rock or kind of poke it with a stick. If it's just from the iron bacteria, it'll kind of break up into platelets on the surface. But if it's petroleum, it'll kind of reconnect. And it might have a petroleum type smell to it. But yeah, if volunteers do see potential pollutants, Kirkland, and I'm sure other jurisdictions want to know about it, because we have professionals that will come out and investigate and try to clean it up if it is a pollutant.
Awesome. Well, thank you. Anybody have any more questions on that one? Okay, I had one more thing I was going to share with you guys, and I'll see if I can get this to work. Let's see. Sorry, I'm gonna do one more thing. This is going to be a little video. So Cascade Water Alliance is one of our partners, and they made a series of videos, and I'm just going to show you one of the short ones. So hopefully this works, just to show you what they came up with recently for us. Hi, I'm Eric Admin. I'm a volunteer with the Sno-King Watershed Council. We're an all-volunteer nonprofit. We train community members. Oh, sorry. Hi, I'm Eric Admin. I'm a volunteer with the Sno-King Watershed Council. We're an all-volunteer nonprofit. We train community members to go out and monitor their local creeks. We appreciate the support that we get from Cascade Water Alliance for our volunteer programs. What we did today is super cool because it is really fun. You're learning a lot, but it's also amazing when you see the community of creatures that live in the bottom of a stream right under your nose that you never were aware of. Found 21 mayflies. One of our program goals is to make people more connected to the streams that they live near, that are part of their community, so they really understand what's affecting that particular stream, and then they can become an advocate for that stream in their community. Tony, I thought those are kind of fun. They made one for biomonitoring, water chemistry, and bacteria, and we did a stream flow and habitat assessment one. So that was a fun one as well.
Eric, can you send the links to that?
Yeah, I'll do that. And I guess just before we wrap it up,
Also, Eric, I wanted to let you know. Sorry, I wanted to let you know that I just put in the chat, or I'm about to put in the chat, it's loading right now, a new booklet that Soundkeeper released called How to Spot and Report Pollution. So it's uploading right now.
Very cool. All right, did anybody else have any other just general questions or issues or stories or anything that they wanted to share while we're here? All right, well,
Eric, sorry.
yeah.
Just a couple things. First of all, you may recall that I was trying to get the Excel files, the Excel data downloaded from the Global Water Watch. Do you know if they ever fixed that error? Did they ever send you any information on that?
I emailed Serge at Global Water Watch about that, because an option through the Global Water Watch database is download to an Excel file, and that was working for the bacteria, but not for the chemistry. So I emailed Serge about that. He was just heading off on a trip to Chile. He said when he got back, which was going to be, I think, this week, he was going to look at that. So I don't think it's fixed yet, but it's on their list. But kind of along those lines, I didn't really have a big database update, but I'll just put a bookmark on. I think that I may be switching potentially to a different scheme of entering and accessing data, because we have had some issues with Global Water Watch software.
And would you have a place to put in stream flow, since we just started to measure our stream flow and volumes? So that would be kind of a neat thing to also have as a data point, a data reference point.
Yeah, I would think that we could have stream flow, and we could also include stream biomonitoring information.
Great.
Okay, well, as always, I just want to say thank you, everybody, and I also want to say we have to thank all of you, thank our partners, thank those people who are paying for our programs, and those people who help us do this work.
Eric, I did have one question.
Yes.
Well, actually, two questions. One is about copper testing. That's kind of an extension to that is, are there any other people, any of the people here doing testing in salmon streams and therefore testing something other than just the basics from GWW? In particular, like I mentioned, copper, I'm thinking we may add copper to ours because ours is a salmon stream. I would love to be able to do 6 ppd, but they don't have a test for that yet. So we already know that the coho die within 24 hours of coming into our stream. So yeah, that's the question.
Good, Dave. Yeah, we do copper and zinc phosphates and nickel. So we do this photometer that Hannah makes, and I think Eric's got one as well. So yeah, and those are in parts per billion in some cases, or parts per million. So yeah, depending. So yeah, we do those kinds of tests.
How much is it?
So we bought it a few years ago, and the photometer, I think, was like somewhere between $400 and $450. The tests themselves, which are little packets, generally they can run, less than $1 a test to do. But you have to buy them. You might have to buy 100 tests, 100 packets in a package. But it's once you got the equipment, it's not overly expensive to do the tests. Some of their tests aren't really as reproducible as I'd like to see them. Like nitrate is one that we have, but I've never, unless the nitrates are high, you really don't get very consistent numbers. Some of the tests are reasonably good, phosphate, copper. So yeah, but it's you make an initial investment of about, it's probably gone up. It's probably more like $600 these days. Who knows the way the prices have gone up, but still it's reasonable.
Okay, well, maybe I'll talk to you offline at some point about the different tests you're running.
Yeah,
sure. I know I've tested, I have some of the testing that I use for aquariums that I've gone ahead and done in our pond. And like nitrates, I've never found high. Ammonia, I've never found high. Et cetera. But yeah, copper, I know we have found high.
Yeah.
And that was a dropper test, a reagent test.
Right. Yeah. No. Yeah. We can talk.
All right.
And then I have, so Anna's link in the chat right now about the how to spot pollution. And then also Anurag had a question about, do we have an organization ID to upload data to the database, the EPA water data? I have to see what sort of organization ID that they require. So I'm not sure what where.
Yeah, so you have to register for that. And I wanted to know if we have registered there as a Global Water Watch or Sno-King, because there was one organization for King County, one for Snohomish County, but I'm not sure if the data that we upload to these local organization if they upload all of our data already in there. So that is something we need to ask people where our data goes to, because this portal is pretty much used by everybody who is going to do modeling here, and that's the easiest way for people to get data access around the world.
Yeah, good question. I don't believe that we already have an ID registered for Sno-King for that website, for that EPA portal.
Okay.
Okay, well, great. Well, thank you all very much for joining in on the meeting and sharing information this evening. And look forward to connecting with you guys some more in the future.
Great. Thank you.
Thank you, Eric. Thank you to all the speakers.
Thanks,
Thanks,
Eric.
Eric.
everyone.
Yeah.